हेलो फ्रेंड्स, वेलकम टू न्यू चैप्टर नंबर 37। आज हम लोग ब्लॉटिंग टेक्निक्स के बारे में जानेंगे। ब्लॉटिंग टेक्निक में हम लोग डीएनए, आरएनए, प्रोटीन ये सब का पता करते हैं। तो चलिए शुरू करते हैं और जानते हैं कुछ नया, कुछ इंटरेस्टिंग। तो चलिए शुरू करते हैं ब्लॉटिंग टेक्निक्स। Blotting is a powerful and sensitive technique for identifying the presence of specific biomolecules within a sample। तो इसमें ये क्या है, एक पावरफुल और सेंसिटिव टेक्निक है जिसमें हम लोग आइडेंटिफाई करते हैं, पहचानते हैं। क्या पहचानते हैं? तो स्पेसिफिक बायोमोलिकुल्स को, जो कि प्रोटीन है, डीएनए है, आरएनए है, ये सब टाइप्स ऑफ ब्लॉटिंग आर डिफरेंशिएटेड बाय द टारगेट मॉलिक्यूल दैट इज बीइंग सॉल्व्ड। The first of these techniques developed was the Southern blot, named for Dr. Edwin Southern, who in 1975 developed it to detect a specific DNA sequences. Subsequently, the method was modified to detect other targets. The nomenclature of these techniques was built around Dr. Southern's name, resulting in the terms Northern blot for detection of RNA, Western blot for detection of protein, Eastern blot for detection of post-translationally modified proteins, and Southwestern blot for detection of DNA binding proteins. The blotting methods are fairly simple and usually consists of 4 separate steps: electrophoretic separation of protein or of nucleic acid fragments in the sample, transfer to and immobilization on paper, nitrocellulose (PVDF) support, binding of analytical probe to the target molecule on paper, and visualization of bound probe. Blotting techniques are selected based on the target molecule DNA, RNA और प्रोटीन। तो 3 तरह का ब्लॉटिंग टेक्निक है मेनली। साउदर्न ब्लॉट, जिसमें डीएनए, टारगेट मॉलिक्यूल डीएनए होता है, सैंपल प्रिपरेशन, डीएनए एक्सट्रैक्शन, एंजाइमेटिक डाइजेशन होता है, सेपरेशन, इलेक्ट्रोफोरेसिस, मेम्ब्रेन मटेरियल, नाइलॉन प्रोब, न्यूक्लिक एसिड प्रोब विथ सीक्वेंस होमोलोगस टू टारगेट, प्रोब लेबल, रेडियो लेबल, एंजाइम डिटेक्शन मेथड, एक्सरे फिल्म, केमिल्यूमिनेसेंस। दूसरा है नॉर्दर्न ब्लॉट, जिसमें टारगेट मॉलिक्यूल आरएनए होता है, सैंपल प्रिपरेशन, आरएनए आइसोलेशन होता है, सेपरेशन, इलेक्ट्रोफोरेसिस, मेम्ब्रेन मटेरियल, नाइलॉन प्रोब, आरएनए, डीएनए और ओलिगो डीऑक्सीन्यूक्लियोटाइड, प्रोब लेबल, रेडियो लेबल, एंजाइम डिटेक्शन मेथड, एक्सरे फिल्म, केमिल्यूमिनेसेंस। तीसरा है वेस्टर्न ब्लॉट, टारगेट मॉलिक्यूल प्रोटीन, सैंपल प्रिपरेशन, प्रोटीन एक्सट्रैक्शन, सेपरेशन, इलेक्ट्रोफोरेसिस, मेम्ब्रेन और मटेरियल, नाइट्रोसेलुलोज और पीवीडीएफ, प्रो प्राइमरी एंटीबॉडी, प्रोब लेबल, एंजाइम डिटेक्शन मेथड, फिल्म कूल्ड सीसीडी, कैमरा, एलईडी और इंफ्रारेड इमेजिंग सिस्टम। अब समझते हैं साउदर्न ब्लॉट के बारे में डिटेल से। Southern blots are used to determine the identity, size, and abundance of a specific DNA sequences. The Southern blot protocol begins with DNA extraction from the cells or tissues, which is then enzymatically digested to produce DNA fragments. The fragments are separated by size on an agarose or polyacrylamide gel via electrophoresis. A smaller fragments will migrate farther on the gel than larger ones. Following electrophoresis, the DNA on the gel is transferred to a nylon membrane. The membrane is incubated with a nucleic acid probe. The membrane is incubated with a nucleic acid probe that has a sequence homologous to the target sequence and is labeled with radioactivity, fluorescent dye, or an enzyme capable of generating a chemiluminescent signal. Hybridization of Complementary sequences occurs during incubation, and the unhybridized probe is removed by washing with buffer. The fully hybridized label probe molecule will remain bound to the blot. Detection methods differ based on the probe label. Radio-labeled probe are visualized with X-ray film or phosphor imaging, and enzymatically labeled probes are visualized with chemiluminescent substrate. Southern blot protocol. First, DNA isolation. Second, restriction digestion. Digest the DNA with a restriction enzyme, and if necessary concentrate digested DNA. Third, gel electrophoresis. Prepare an agarose gel and either TAE or TBE buffer. Buffer selection will depend on the duration of the run and the size of DNA fragments. Load sample into wells and include your DNA molecular weight marker. Run the gel. 4. Transfer. Place the gel in a container with denaturing solution and wash twice for 15 minutes on a shaker. Rinse with water, then wash with neutralization solution. During the previous step begins to prepare Whatman paper and nylon membrane for the transfer. Assemble the transfer apparatus with the membrane, Whatman paper and gel. Transfer in SSC or SSP buffer. When transfer is complete, crosslink DNA in a crosslinker, then rinse the membrane. When transfer is complete, cross-link DNA in a crosslinker, then rinse the membrane. Fifth step, pre-hybridization blocking. Blocking reduces non-specific binding to the membrane. Prepare the pre-hybridization solution and add sample DNA. Remove the blot from the crosslinker, add the pre-hybridization solution and incubate. Sixth, hybridization. Prepare the probe mixture, a complementary DNA strand and buffer. Remove the pre-hybridization solution and incubate the blot with the probe. Incubation times will vary depending on the application. Following incubation, perform a low astringency wash followed by a high astringency wash to refine the DNA. Seventh step, probe detection. Rinse the membrane, transfer to a container with blocking solution and incubate. Discard blocking solution, replace with antibody solution and incubate. Discard antibody solution, wash the membrane. 8th step, follow manufacturer directions for chemiluminescent detection. Application of Southern blotting are used in gene discovery, mapping, evolution and development studies, diagnostics and forensic DNA fingerprinting, preparation of RFLP maps, identification of the transferred genes in transgenic individuals. To study the genetic arrangements, DNA, endonuclease, DNA fragments, electrophoresis, agarose gel, blotting, filter paper, nitrocellulose, agarose, incubation with radio-labeled clone DNA probe, covering with photographic plate, Position of DNA fragments complementary to probe. साउदर्न ब्लॉट, ये हमारा साउदर्न ब्लॉट कंप्लीट हुआ। अब दूसरा है नॉर्दर्न ब्लॉट, जिसमें हम लोग आरएनए को डिटेक्ट करते हैं। नॉर्दर्न ब्लॉट्स आर यूज्ड टू डिटरमाइन द आइडेंटिटी, साइज एंड abundance of a specific RNA sequence. Northern blot protocol was developed by Alvin. It begins with RNA isolation and separation techniques vary depending on RNA size. Large RNAs are separated by electrophoresis on a formaldehyde agarose gel. Large RNAs are separated by electrophoresis on a formaldehyde agarose gel or glyoxal agarose gel, which prevents normal base pairing and maintains RNA in a denatured state. Small RNAs are separated on a denaturing urea polyacrylamide gel. The RNA is then transferred from the gel to a nylon membrane. The RNA is then transferred from the gel to a nylon membrane, which is then incubated with a radioactively or non-isotypically labeled RNA, DNA, or oligodeoxynucleotide probe. Okay, the RNA is then transferred from the gel to a nylon membrane, which is then incubated with a radioactively or non-isotypically labeled RNA, DNA, or oligodeoxynucleotide probe. The unhybridized probe is removed by washing with buffer. Radio-labeled probes are visualized with X-ray film, and enzymatically labeled probes are visualized with chemiluminescence. Northern blot protocol. RNA isolation. First step, RNA isolation. Second, electrophoresis. For a formaldehyde agarose gel, prepare the gel and insert the gel tray into the apparatus. Fill with MOPS buffer. Load the sample and include a molecular weight marker. Run the gel, then trim the gel prior to blotting. For a glyoxal agarose gel, prepare the gel and insert the gel tray into the apparatus. Fill with MOPS buffer. Prepare sample and load into wells along with RNA ladder. For a denaturing polyacrylamide gel, cast the gel and mount it in the electrophoresis unit. Prepare sample, load into the gel and run with TBE running buffer. Third step, transfer. For a formaldehyde agarose gel or glyoxal agarose gel, wash the gel in SSC, then assemble the transfer unit with the gel, filter paper and nylon membrane. When transfer is complete, place the membrane in a UV cross linker. For a denaturing polyacrylamide gel, assemble the transfer unit including gel, filter paper and nylon membrane, ensuring they are flooded with TBE. Then when transfer is complete, place the membrane in a UV cross linker to fix the RNA to the membrane. Pre-hybridization blocking. Pre-hybridized the membrane in hybridization solution. Fifth step, hybridization. Add probe to the hybridization solution and incubate. Wash the membrane in low astringency washes to remove hybridization solution and unhybridized probe, and high astringency washes to remove partially hybridized molecule. हाइब्रिडाइजेशन में क्या करते हैं, एड प्रोब टू द हाइब्रिडाइजेशन सॉल्यूशन एंड इंक्यूबेट कर देते हैं. फिर वॉश द मेम्ब्रेन इन लो स्ट्रिंजेंसी वॉशेस टू रिमूव हाइब्रिडाइजेशन सॉल्यूशन एंड अनहाइब्रिडाइज्ड प्रोब एंड हाई स्ट्रिंजेंसी वॉशेस टू रिमूव partially hybridized molecule. Follow manufacturer direction for chemiluminescence detection. Application of northern blotting. क्या एप्लीकेशन है, तो पहला, या स्टैंडर्ड फॉर द स्टडी ऑफ जीन एक्सप्रेशन, या स्टैंडर्ड फॉर द स्टडी ऑफ जीन एक्सप्रेशन एट द लेवल ऑफ मैसेंजर आरएनए ट्रांसक्रिप्ट्स. सेकेंड, डिटेक्शन ऑफ मैसेंजर आरएनए ट्रांसक्रिप्ट्स साइज. Third, a study of RNA degradation. Fourth, a study of RNA splicing. And fifth, a study of RNA half-life. Next is Western blot. Western blotting technique was first developed by W. Neil Burnett and is used to determine the identity, size, and abundance of a specific protein within a sample. The Western blot protocol begins with sample lysate preparations from tissue or cell culture and separation on a polyacrylamide gel via electrophoresis. The separated proteins are then transferred to a nitrocellulose or polyvinylidene difluoride PVDF membrane. The membrane is incubated with a blocking agent to prevent non-specific binding, followed by incubation with a primary antibody to bind the protein of interest. There are two detection methods, direct or indirect. Direct detection relies on a labeled primary antibody, whereas indirect detection requires a primary antibody directed against the target protein. And a secondary antibody directed against the immunoglobulin class or subclass of the primary antibody's species. Visualization method include colorimetric assay in which a colored precipitate is produced, chemiluminescence and fluorescence. Western blot protocol. First, prepare lysate from cell culture or tissue. Second, sample preparation. Determine the protein concentration of each sample with a protein quantification assay, that is Bradford assay. Add a equal volume of 2x Lamley sample buffer to each sample. Some samples may need to be reduced or denatured. This is achieved by boiling samples in a buffer. Third, electrophoresis. Prepare an SDS page gel. Load samples along with molecular weight marker. Run the gel in running buffer. Fourth, transfer. Following electrophoresis, assemble the transfer unit including the gel, PVDF or nitrocellulose membrane and filter paper. Transfer the proteins to the membrane with transfer buffer. Fifth, antibody staining, indirect detection. Prepare blocking buffer and incubate the membrane to reduce non-specific binding. Second, incubate the membrane with primary antibody diluted in blocking buffer. wash the membrane in TBST and incubate with conjugated secondary antibody dilution in blocking buffer. Okay, antibody staining or indirect detection. First, prepare blocking buffer and incubate the membrane to reduce non-specific binding. Second, incubate the membrane with primary antibody dilution in blocking buffer. Third, wash the membrane in TBST and incubate with conjugated secondary antibody diluted in blocking buffer. Fourth, wash the membrane in TBST. And fifth, follow manufacturer directions for chemiluminescent detection. Application of Western blotting to determine the expression of protein. To determine the expression of protein, to measure the efficiency of gene knocking down using siRNA, to check the presence or absence of a protein in disease, to determine the efficacy of drugs. To determine the efficacy of drugs. Next is Eastern blotting. Eastern blotting technique was given by Bogdanov. This method is used to identify carbohydrate epitopes including glycoconjugates and lipids. यह है ईस्टर्न ब्लॉटिंग. और एक है साउथ वेस्टर्न ब्लॉट. इट इज बेस्ड ऑन द साउदर्न ब्लॉट एंड was first described by B. Bowen, J. Steinberg and colleagues in 1980. The name southwestern blotting is based on the fact that this technique detects DNA binding proteins. Since DNA detection is by southern blotting and protein detection by the western blotting, it is a lab technique which involves identifying and characterizing DNA binding proteins by their ability to bind to a specific oligonucleotide probes. The proteins are separated by gel electrophoresis and are subsequently transferred to nitrocellulose membranes, similar to other types of blotting. The proteins are separated by gel electrophoresis and are subsequently transferred to nitrocellulose membrane, similar to other type of blotting. यहाँ फिगर में दिखाया गया सैंपल मिला, उसमें क्या किए, एक्सट्रैक्शन किए डीएनए आरएनए प्रोटीन, फिर इलेक्ट्रोफोरेसिस से हम लोग सेपरेशन किए. उसके बाद अ स्मॉलर मॉलिक्यूल विल माइग्रेट फार्दर डाउन द जेल. फिर मेम्ब्रेन ट्रांसफ़र, उसके बाद लेबल्ड प्रोब इंक्यूबेशन, उसके बाद टारगेट डिटेक्शन. फिगर 96 ब्लॉटिंग इन नट सेल. ओके फ्रेंड्स, ये चैप्टर कंप्लीट हो चुका है. नेक्स्ट चैप्टर है 38. टॉपिक है एसडीएस पेज इलेक्ट्रोफोरेसिस. इसे हम लोग नेक्स्ट वीडियो में कवर करेंगे. तब तक के लिए बाय बाय. हंसते रहिए, मुस्कुराते रहिए और पढ़ाई करते रहिए. बाय बाय.